Hello everyone, welcome to the next lecture of week 2 in which we are discussing about meristematic tissues, which are basically dividing cells and are responsible for giving all different cell types and tissues which are found in a plant organ. Now in this lecture we will be talking about shoot apical meristem. Now apical meristem, as we have discussed before, that they are sites of primary growth, means apical meristem give rise to the primary tissues which are present throughout the growth of an organism, and they increase the height of the plant and length of the plant. They are found at terminal position and they are also responsible of forming the lateral organs of a plant. And these are found in shoot apical region and root apical region known as shoot apical meristem as well as root apical meristem. Now in this lecture we will talk about shoot apical meristem, and in the next lecture we will discuss about root apical meristem. Now shoot apical meristem, as we have discussed before, that they are found on the growing points of a shoot. Okay, so this all we have talked about. At the growing points, at the branches also, these shoot apical meristem is present. Okay, so basically they are terminal in position, means they are found at the tip of the plant. And these apical meristem has unlimited capacity of growth and they can divide continuously to give rise to new tissues or organs. So we can say that they are a reservoirs of stem cell, means those cells which have the ability to differentiate into different tissues and organs. So from there they will divide and finally they will get differentiated into other different types of tissues. Now shoot apical meristem arises from, give rise to all above ground organs of a plant, means all the things which are present on the shoot arise from shoot apical meristem. So that all we have discussed, like leaf, stem, branches, flowers. So they all come out from the shoot apical meristem, as well as some underground portions also, like stem like tubers, but they are also actually they originate from shoot apical meristem. Okay. Now, if we talk about the structure of this shoot apical meristem, they are dome shaped. So if we assume this is the structure of a growing shoot and this is the tip of the shoot, so at this point you will have shoot apical meristem. And you can see here that lateral appendages are also coming out. You will have leaf primordia. Okay, they are all coming out from this leaf, and this is all growing young leaf. So these are all coming out from this shoot apical meristem. This is a small axillary bud. Now when we observe it carefully, we can see here that this shoot apical meristem, actually they will give rise to different tissues or different layers which will form different parts of the plant. They shoot apical meristem they will form protoderm, then ground meristem and procambium. You can see here that this is the outer layer, protoderm, okay, which will form the epidermis. Protoderm will give rise to epidermis, okay, and then you have ground meristem, which will give rise to cortex, pith, okay, other parts of the plant, and then you have procambium, which will give rise to vascular tissues. Okay. So this is the organization of shoot apical meristem. However, in detail also we will discuss that what is the organization, and the size of the shoot apical meristem can vary. Actually, it is a very small region. Now, the size can vary from 120 to 300 micrometers. The size can be small also, and it has also found that a very big size of 1000 micrometer has also been observed in some plants. However, it can vary depending on the species of the plant as well as depending on the age of the plant. The size can vary. Similarly, the height of this apical meristem can also vary depending on the age of the plant, depending on the species. Now, this we have discussed that this will give rise to protoderm, procambium and ground meristem, and lateral organs such as leaves, axillary bud, flowers, lateral branches, they all will come out from the shoot apical meristem. Now, it's important to note here that these lateral appendages which are coming out, they are exogenous in origin, means they are originating from the outer layers. It is not outer layer. Okay, it's not that some certain cells present on the inner side of the plant body is giving rise to these lateral appendages, and that we will see there is a difference between shoot apical meristem and root. So, in that cases we will see that root apical meristem is actually not giving rise to any lateral organs. Okay, however, the shoot apical meristem is giving rise, and their origin is exogenous, okay, from the outer layer or from the outer surface. Now, when the meristem is developing, okay, suppose it is developing some axillary bud, okay, suppose at certain point some axillary bud is getting formed. So, in this case also you will see that this axillary bud will also develop its apical meristem. Okay, they will also start developing its apical meristem, and they can form later some branches of the plant. Now, when we talk about the organization of this shoot apical meristem in vascular plant, so now it's not about only angiosperm. All the vascular plants, means all those plants which has vascular system for conduction, even pteridophytes are also included in that, gymnosperms are also there, and higher flowering plants are also there. So, in vascular plants, basically two categories of these shoot apical meristem have been identified. Unicellular means shoot apical meristem containing only one cell, okay, only one meristematic cell is there, which is mostly found in pteridophytes, and definitely multicellular, in which different group of cells are there which perform the function of apical meristem. Now, this pteridophyte, how it is done, we will deal in this detail later when we will talk about theory. So, that time also we will try to understand how a single cell is giving rise to entire part of the plant. And in higher plants you have multicellular organization, in which definitely flowering plants will come. Now, this is just a picture to show pictures where you can see shoot apical meristem, you can see a growing shoot apical meristem, you can see axillary buds are there which will also later form their own meristem, and in the top you have a dome shape shoot apical meristem, and these are the leaf primordia. and vascular strands are also there. Then you have, these are the vascular tissues, okay, vascular tissues. Then you have cortex, and then you have epidermis, okay. So you have basically protoderm, it's actually protoderm, okay, protoderm. This is ground meristem, and these are the procambium, okay. Now this picture shows that how different pattern of you can say phyllotaxy, or different arrangement of these lateral appendages takes place in a shoot. So you would have seen different arrangement of leaves which are oriented in different ways on a branch or on a shoot, like you say lateral arrangement, opposite arrangement, something like that. So this all is arranged depending on how the shoot apical meristem is forming the leaf primordia, and this is all genetically controlled, okay, what type of pattern or what type of phyllotaxy the leaf will have on the shoot. Now coming to a more detailed structure of shoot apical meristem. Now, when we will observe more closely the dome shaped structure, actually that is the shoot apical meristem. So, you will find different layers, okay. Now, different layers, like these layers are designated as L1, L2 and L3. See, variations are all there, but here I am just trying to talk the generalized structure which is found in angiospermic plant. So, 3 layers will be there, and they will be divided on the basis of how they are dividing, or like means in which plane they are dividing. Now, let us try to understand the structure of this layering. Okay, so you will find 3 layers. This is the L1 layer, okay. The second is the L2 layer, and the third is the L3 layer. Now you can see here that L1 and L2 layer, you will see they are dividing in anticlinal fashion, okay. They are at right angles to the surface, okay. To the surface they are dividing in this way, suppose perpendicular to the surface. So, in that case they will increase the surface of the layer, okay. They are not increasing the volume. So, L1 and L2 layer will also divide in anticlinal division, and they will actually give rise to the outer surface. Mostly L1 layer will give rise to the outer surface, L2 layer will also contribute, and many a times it contributes to the inner part of the cells also, and the L3 layer will divide it all planes, as you can see here that it is dividing it all plane, and it is contributing to the overall increase in volume of the plant. Now, another way is also there, when a close observation was was done for this meristematic region on the basis of cytohistological studies, different zones were observed. Now, cytohistological studies means study of the internal organization of cells, you can say. Now, how the cells are behaving, how densely their chloroplast is there, what is the nature of their metabolic activity, how they are dividing. So, that will give them some change in the histological character of that layer. So on that basis also different zones have been observed. Okay, so when this cytohistological study was done, different zones was observed, in which you have a central zone, then you have organizing center, and then you have peripheral zones. This is the peripheral zone and this is the rib zone. Now central zone, as you can see here, that it is a zone of dividing cells, and it is present in the top position, and a small area is occupied by the central zone, and in this zone a small group of cells are present here which are not as actively dividing as compared to other zones of this layer. Then you have an organizing center and the peripheral zones. On the peripheral side, these are also known as flank zone, and the rib zone in the center, which occupies the central position, and it gives rise to different parts of the plant. Now central zone, see, as this is a slowly dividing stem cell, so basically these will be very low in their DNA and RNA content, they will be thick walled, they will be highly vacuolated and large, the cell size will also be large. Then plastids will be undifferentiated and their mitotic index, means the division rate, will be very low as compared to other zone. However, it is considered that these central zone will give rise to new cells of peripheral and rib zone, and they are a reservoir of pluripotent stem cells, okay, because giving rise to different organs of the plant. Now you have peripheral zone, okay. These cells, their growth rate is very high, okay, and they have higher DNA content, they have higher RNA content, they have thin wall. So these peripheral zones will divide and they will give rise to peripheral organs like axillary buds, leaf, and peripheral parts of the stem will come out from this peripheral zone, and then the rib zone, this will give rise to pith of the plant, means the pith and cortex of the plant will come out from the rib zone. Now, a small organizing center is also there, present just beneath the central zone. Actually, organizing center is required to induce and maintain the stem cell fate. Some sort of signals and some sort of chemicals will be secreted from organizing center, and they will regulate the fate of these stem cells, that which particular stem cell will lead to which particular organ. Suppose if any sort of stem cells are lost in the peripheral zone or the rib zone because of any activity, they will be filled up by the cells which are coming out from the organizing center. Now, these shoot apical meristem and the cells which are present in the shoot apical meristem are controlled by various genes and various hormones. There are some sort of genes which are responsible to control the fate of these stem cells, as well as different hormones are there which regulate the fate of these stem cells, among which auxin is very important hormone. Auxin is also required for stem elongation, okay, and it also required for differentiation of and physiological function of various cells which are present in the shoot apical meristem, and they actually trigger primordia cell differentiation, okay, all the lateral organs, leaf primordia which is coming out. So, they trigger their differentiation and control the patterning properties of the shoot apical, means in which pattern the primordia will come out and how they will be oriented, that is all controlled by auxin. Similarly, gibberellins are also important growth hormones. They also promote initiation of lateral organs along with auxin. Now, high level of cytokinin is also found to be useful in maintaining the reservoir of these stem cells that enable indeterminate. They are also found to be responsible for maintaining the population of these stem cells. Okay, so for today we have discussed about the structure and how the stem cells are organized. In the next lecture we will deal about the root apical meristem and how they are organized. So, thank you for today.